Okay, fine. Just wait for 1 2 minutes and then we'll start the session. Okay, so good evening, everybody. So today we'll be discussing questions which are related to week 3 uh, of the developmental biology course. So, as we have studied in our previous week class, we completed the differential gene expression portion of this particular course, and then we started off with the genetic basis of basic genetic concepts which are important and will be useful for our this subject, developmental biology. We studied about some of the cross. We studied how to solve questions related to probability. Yeah, basic probability questions, and also important question which was related to 3 point test cross, which is an important question which often comes into very much in examinations, in your competitive examinations in CSIR and all. So that is an important concept. So today we'll be continuing what we were discussing in the last week, that is the genetic basis and the basics of genetics, which are important for your developmental biology subject. So, as we always do, we'll start with the questions and we'll try to learn the concepts which are related to that particular questions. Okay. So, okay, fine. So, we'll start with the questions. So the first question is, which of the following chemicals can be used to induce mutation? EMT, EMS, MET, and MES. This is the question. I'm repeating it again. Which of the following chemicals can be used to induce mutation? EMT, EMS, MET, and MES. 2 minutes to answer this question and then I will switch the screen. Okay. Just a minute. I think there is any pop up. I have to see whether someone wants to join or is there any problem. Okay. No, okay, fine. Okay. So I'll request that please wait for 2 minutes and then answer. Even if you know the answer, just wait because it will help others also to think and answer. But you are answering, it's good. Okay. So let us see what you have answered. So I can see that only Ajay has answered. Others, why you are not answering? Try to at least try. At least try to select one option. Hmm. Okay. So Shubhamoy and Ajay has answered B, which is EMS. So can anybody tell me what is the full form of EMS? What is EMS? What is the full form of EMS? EMS stands for ethyl methane sulfonate. Okay, ethyl methane sulfonate. Just a minute. I'm sorry, I'm not very well today. So maybe that's why my voice is not that loud enough. Okay. So the correct answer is EMS. So EMS, it stands for ethyl methane sulfonate. Okay. So basically what it does is that it is an alkylating agent. I mean, it induces mutagenesis by alkylating the guanine bases which are present in the DNA. So it leads to base mismatching. It leads to base mispairing. So if you I won't go into that much detail about the mutation. I mean the mutation because it itself is a very vast topic. But just for a brief overview, so basically mutations can lead to various kinds of transitions like GC to AT transition is one of the most common transition that occurs during mutation. Okay, that is GC to AT transition. So this particular mutagen, so what is a mutagen? Chemical or an agent that causes mutation, that is known as a mutagen. So this EMS, which is ethyl methane sulfonate, it is a mutagen and it causes base mispairing or base mismatching, which results into GC to AT transition. Remember this, that EMS causes GC to AT transition. And so basically what happens, that this uh, EMS, it reacts with the ethyl group and uh, it forms an abnormal base which is known as ethyl guanine, O6 ethyl guanine. It forms an abnormal base which is known as O6 ethyl guanine, and this ultimately leads to GC2AT transition. Okay, now we'll go to the next question. I'm sorry. I think you saw the answer. Anyways, so the next question is, a mosaic animal will contain wild type cells, mutant cells, both A and B, or only mutant cells? Option D. Think what can be the answer? Just one more minute and then I will switch the screen. Okay, let us switch the screen. See what you have answered. Okay, Ajay. Okay, just why it's showing it again. Anyways, okay. B, sorry, C. So Ajay has answered C, Kartika has answered C, Jasrina has answered C, and Shubhamoy has also answered C. So what is the C? Both A and B. So all of you have answered it correctly. So a mosaic animal will contain both wild type cells and mutant cells. Okay. So now we'll look a little bit about what is mosaicism. Mosaicism, because here we are studying about genetics or developmental biology. So we should study this in context to what we are studying, that is genetics. So if I talk about hmm, genetic mosaicism, it is basically, what does it means? It means presence of two or more cell lineages with different genotypes which are present in a single zygote. Okay, so one thing which is important when we study about genetic mosaicism is that we are talking about a single zygote. First thing, it is important. Okay, and another thing which is important is presence of two or more cell lineage. Presence of two or more cell lineage, and these lineages have different genotypes. Okay, so this is your mosaicism. That means two or more cell lineages are present in a single zygote. This is your mosaic condition. Then what is a chimera condition? Chimera or chimera, whatever you want to call it as. Chimera. What is your chimerism then? Because why I am stressing on these two terms? Because students often gets confused between a mosaic and a chimera. That means mosaicism and chimerism. So that's why, does anybody know what does chimerism means or what is a chimera? Anybody? So Ajay has answered two species. Okay. Anybody else who would like to answer? Just Reena, Kartika, Shubhamoy, anything else that you can could think of? So think cell line in one organism. Okay. So to differentiate between a mosaic and a chimera, in the simplest term, okay, if I write here mosaic and a chimera, okay. So in mosaic, we talked that different genotypes, that means cells from different lineages, which are arising from a single zygote in a single individual. So here we talked about a single zygote. Okay, two or more different lineages. Whereas when we talk about chimera, basically these are cell lines which are derived from different zygotes. Okay, cell lines which are derived from different zygotes. That means this mosaic, it is a post zygotic mutation. Would you understand? So basically chimera means that distinct cell lines which are derived from different zygotes, that term is known as chimera. Whereas mosaicism is a term which is defined as that there are two or more lineages, cell lineages which are present, which has a different genotype which are arising from a single zygote. So this is the basic difference. Okay, fine. Now we'll come to the next question. If the central if the central VPC is destroyed, what would happen to the development of primary vulval cells? A. They will not be formed. B. They will transform into hypodermis. C. They will be formed normally. Or D. They all will acquire tertiary fate. Try to think and answer this question. 2 minutes for this question. 1 minute more. Okay. Okay, I'm switching the screen now. Hmm. Hmm. So Ajay has answered C, that is, they will be formed normally, and Shubhamoy has answered C. C, they will be formed normally, and what is the correct answer? C. So C is the correct answer. So when we talk, so I think this vulval cell, the uh, for this in much detail, I think sir has described in the lecture. You can go and you can see through the lecture. I'll just try to brief it out that what is VPC? VPC stands for vulval precursor cells, VPC. So basically in C. elegans, the development of vulva, it involves a series of interaction between different cells, particularly the anchor cells and the vulval precursor cells. Okay. So there are many factors which are involved. Here we'll concentrate on, suppose we say lin 3, lin 12. So there are many factors involved. And lin 3, it is a gene which encodes a ligand which acts on the vulval precursor cell and it induces your vulval development. Okay. This is like the basic background information that is needed to understand the question. So now the central part of the VPC, the central VPC, it receives the highest level of link 3 signal. Okay. So the central portion, central region, it receives the highest level of lin 3 signal and it becomes the primary vulval cell. So from this we can say that primary, secondary and tertiary fates, it depends on what? It depends on the levels of lin 3 that they receive. So primary fate would be adopted by the central portion and it becomes the central part of the vulva. Secondary fate is adopted by the neighboring VPCs. That means if this is the central region, the neighboring regions, which is mostly P5 or P7. So these regions becomes the secondary fate. So this is the primary fate because it receives the highest amount of Lin3 signal. This becomes the secondary fate because it receives a lesser amount of Lin3 signals. And tertiary fate is adopted by the other verbal precursor cells that do not receive the enough Lin3, and it can differentiate into hypodermis because they do not receive the correct level of Lin3. Okay, now if we look into the question says that if the central verbal precursor cell is destroyed, I mean the central, this is the region, it is destroyed, what would happen to the development of primary verbal cells? So now by the concept we know that the central region, it receives the highest level of Lin3 and it becomes the primary verbal cells. But the central region is destroyed. So what will happen? Will they not be formed? No, they can be formed. Why? Other vulval precursor cells, they might compensate and it can adopt for the primary fate. So this is not correct. They will transform into hypodermis. So, if the central area, the central verbal precursor cell is destroyed, the fate of the primary verbal cells, it might be adopted by another VPC rather than it get transformed into the hypodermis directly. So, this is also incorrect. They will be formed normally. Yes, they will be formed normally because the other VPCs, they can compensate and they can adopt for the primary fate, and they will acquire tertiary fate. uh, this is tertiary fate acquire, no, it cannot acquire tertiary fate because the loss can be compensated by the other verbal precursor cells. So the correct answer here would be C. Okay, this is just matching, nothing much to explain. Find the correct match. You just have to tell me the correct answer. There's nothing to explain in this question. I'll give you 2 minutes to solve this question. One minute more. Okay. Now I am switching the screen. Hmm. Hmm. So Ajay has answered sixth, and okay, only Ajay has answered, no one else. A and C. Let 23, lane 3, and lag and lane 12. Yes, these are the correct answers. So this is correct. A and C is correct. There's nothing much to explain. You just have to go through the facts. It's not any concept here. Now we'll move to the next question. Hmm. Which of the following can be used to infer whether two mutations that give the same phenotype affect the same gene. Okay, listen to the question correctly. Which of the following can be used to infer whether two mutations that give the same phenotype affect the same gene? Back cross, complementation, epistatic analysis, genetic mapping, and test cross. 2 minutes. Uh, Someone has kept the mic unmuted. Can you please mute your mic? I'm again repeating the question because earlier there was little noise. The question says, which of the following can be used to infer whether two mutations that give the same phenotype? So there are two different mutations, and those mutations are giving the same phenotype. Whether these two mutations affects the same gene or not. Backcross, complementation, epistatic analysis, genetic mapping, and test cross. Hmm. Think. Okay, now I am switching the screen. Okay, so the correct answer is B, complementation. Yes, Shubhamoy, you can ask. You have raised your hand. You can write your query also. Whatever query you have, you can write it in the message box. Okay, Shubhamoy, you have raised your hand. That is why I am telling. Okay, so okay, B that is complementation. Yes, correct. B is the correct answer. So can anybody tell me what is complementation? What is the meaning of complementation? Complementation is derived from the word complement. Complement means to complete. That means complementation is a type of a mechanism where genes which are complementary genes, it is a kind of gene interaction where both the genes, I mean the two different genes, they work together to create a specific phenotype, specific visible phenotype. Okay, so that means complementation is a kind of gene interaction, and the genes that show this kind of interaction, they are known as complementary genes. And these complementary genes basically work together to give a specific phenotype, and all those genes that work together to give a specific phenotype, they belong to one complementation group. So these are the complementary genes. Okay, and it is a kind of gene interaction. Complementation is a kind of gene interaction. So complementary genes, you can take complementary gene as the genes that helps to complete each other to give a visible phenotype. So here the question says, which of the following can be used to infer whether two mutations that give the same phenotype affect the same gene? So that means these are two mutation. Let's say there is one mutation A and another mutation there is which is B. Now this is a gene X. Okay, this is a gene X. What they are saying that these two mutations they're the two mutations that give the same phenotype. These two mutations are affecting the gene X. That means whatever phenotype this gene X is showing, it is showing because it is affected by both A and B. So A and B together when they work, then they show the phenotype that is uh, the mutation that is there in gene X. That means A and B works together to give the desired or the respective phenotype to the gene X. That means A and B belong to the complementary group. So what we can get to know about complementary genes from here? First point, we can say that complementary genes are two or more different genes that work together to contribute a particular trait. Okay. So if we talk about complementary genes, huh, so complementary genes are two or more different genes that work together. First point. Second, what we can say that complementary genes are simultaneously expressed in the phenotype. Okay, these are simultaneously expressed in the phenotype. Then these genes, basically complementary genes, mix together to produce a distinct phenotype, and they inhibit the production of complement proteins. Complement genes can inhibit the production of complement proteins. These 2 points are important. Okay. Yes. Yeah. So Shubhamoy, you have a query? Ajay, yeah, Ajay. So Ajay, you have given what you have given the whatever examples you have given, they are correct. If any other if there's anything else, you can just write it down. Hmm. Just a minute. Actually, I'm not very well, so it's very difficult for me to speak continuously for 2 hours. Anyways, okay. So the next question is, a mutation in the cis regulatory region of the alpha chain of hemoglobin allows it to be transcribed all the time. This is a type of dash mutation. Which kind of mutation it is? So question is saying a mutation in the cis regulatory region of alpha chain of hemoglobin. So there is a mutation in the alpha chain of hemoglobin, and because of this mutation, the transcription is happening all the time. Okay, there is a continuous transcription, so there is increased transcription. Okay, I'm giving you the hint also. So this is a type of hypermorphic mutation, dominant mutation, gain of function mutation, antimorphic mutation, or a synthetic mutation. Think and answer. Okay, one more minute and then I will switch the screen. Okay, now I'm switching the screen. Hmm. So, okay, so I have got many answers. So, Ajay has answered A and C, A, B, and C. Okay. Jasrina has answered A, B, and Shubhamoy has answered A. Okay. So, A, B, and C. So, the correct answer is all A, B, and C. It can be hypermorphic also, it can be dominant also, and there can be gain of function also. Now, I will explain how and why. So, hypermorphic. What is the meaning of hypermorphic? Hypermorphic means that there is an increase in the normal function. So, whatever is the normal function, there is an increased normal function. Okay. So, if I talk what is hypermorphic, it means there is increased normal function. Dominant means that it if there are two different alleles, the allele which is dominant will always show its function or always show its characteristic, and gain of function again it is related to hypermorphic only. So the question here says that a mutation in the cis regulatory region of the alpha chain of hemoglobin allows it to transcribe all the time. So if this is your alpha chain, okay, and this is the region where the mutation has occurred. Okay, so this is the region where mutation has occurred. Now earlier in normal condition it used to transcribe only for a specific period of time. Now because of this mutation, the transcription is happening continuously. There is a continuous transcription. That means the rate of transcription has increased. That means there is when the rate of transcription has increased, the ultimately translation will also increase, protein production will also increase. So there would be increase in the normal function. There is whatever was the normal function, that transcription is happening. If suppose normal in the normal case, if 10x transcription was happening, now let's say 1000x transcription is happening because of this mutation. So there is increase in the normal function, and it is a dominant because it is showing its effect. That means it is a dominant mutation. And there is a gain of function because whatever the function it has gained some property, that this function is now increased, so it is a gain of function. So therefore A, B, and C all are correct. Okay. Now next question is a very interesting question. I will give you 3 minutes for this question. Okay. So the question says a pea plant heterozygous for the gene P and Q was test crossed and the following progeny was obtained. Assume P and Q are dominant alleles and P and Q are small p and small q are the recessive alleles for these two genes. Calculate the recombination frequency based on two-factor analysis. Try to solve this question. It's a very interesting question, and if students who were present in the last class, I have explained in detail about the 3 point test cross. So if you could understand the 3 point test cross, it is easier than that. Okay, it's way more easier than the 3 point test cross. So try to solve it, and in the text, in the chat box, do not just write the answer, but like just in one line or two, write the explanation of how you have solved the question. Okay, so I'll give you 3 minutes and your time starts now. This table is very important. See all the numbers that are given in the table. This will help you to solve the question. Okay. Sorry. Hmm. Okay, I'm switching the screen now. Okay. Hmm. Yeah, I'm suffering from some viral fever since 1 week. That's why I'm not able to speak properly. Anyways, so Ajay has answered it correctly. 7.4% is the correct answer. So now I will explain this question to you. Hmm. So the question says a pea plant heterozygous for genes P and Q were test crossed and the following progeny was observed. P and Q are the dominant alleles and small p and small q are the recessive alleles of these two genes. Calculate the recombinant frequency based on the two-factor analysis. So whenever we are doing any recombination frequency kind of questions, just a minute. Ah, okay. Hmm. So whenever we are doing any recombination frequency based questions, the very first step, I will tell you this by stepwise method. Okay. So step one is identify the parental and recombinant classes. Okay, whenever you are solving such questions, whether it is a 3-point test cross or it is a 2-point test cross, the very first step is to identify the parental class and the recombinant class. Now if you look into the question, here the question says a pea plant heterozygous for genes P and Q. That means what is the genotype? It is a heterozygous, so it is PPQQ. Okay, was test cross. Now what is a test cross? Test cross is a homozygous recessive. Okay, that means it was crossed with PPQQ. So a pea plant heterozygous for genes P and Q, that means this was one of the wild type which was heterozygous for P and Q, was test cross. That means it was crossed with its homozygous recessive form. So this is your wild type classes. Now let us see, are these, now you can identify wild type classes by two ways. First way, if this table is not given to you, suppose this table is not there, then how can you identify? You can just like how I just solve this, you can also try to solve and make by your own. That is, heterozygous means it would be ppqq, and test cross means homozygous recessive. That means both the alleles are recessive. Recessive genotype is present in homozygous condition, that is your test cross. Now through this table, how we can identify? Last time also I told you that the classes which has the highest numbers, which has the highest number of I mean whatever it is, individuals or plants, whatever it is, which has the highest numbers, those classes are the parental classes. So from this figure, immediately you can tell this, these two are the parental class. Okay, and these two are the recombinant class. Okay, now since it's a two-factor analysis, so single crossover and double crossover, that concept is not required here to understand right now. Now we know these are the parental cross and we are the recombinant classes. Now we have to see that it's very easy actually, but I am telling you in detail just because this detailed explanation will help you to build your concept. Okay, otherwise just you can add these two values with the whole and you will get the answer. But to understand the concept, it's important, because this is a simple question, but what if in your assignment or in your exam a little twisted question is given to you, then you wouldn't be able to answer if you do not know the basics, if you do not know the concept. That is why I'm telling you very slowly so that your concept is clear regarding these test crosses, because last week also there was a question related to 3-point test cross, and this week also there is a question related to 2-point test cross. I mean, two factor analysis. That means that this topic is important. Okay. Now, now you see, these are the two recombination classes which are given. P, P, QQ and PPQQ. Okay, just there's no need of, in this question there is no need of comparison, but just for your understanding per se. Now can you compare the recombination classes with the wild type, and can you tell that the crossover has happened between P allele, I mean P gene or Q gene? Can anybody tell me? It is not important in 2 point test cross. This concept that where the crossover has happened, it is important in 3 point test cross. This concept is not important in 2 point test cross. It's not required. But just for your concept building per se, can you tell me that which gene, whether P or Q, at which gene crossover has happened by comparing the recombinant classes with the wild type class? Can you tell me? Hmm. Mm, Just a minute. It has happened in Q gene. How it has happened in Q gene? See, you compare this with this. P and P are the same, but QQ, this QQ resembles that of this, isn't it? See, this PP is same, but this QQ is resembling of this second parental, this second recombinant class QQ. So that means there is a change at QQ here. Here also see, this is small p. See, this is small p. Here also you see small p, it is same, but here should be small q small q. So that means there is an exchange of this and this. Okay? That means recombination has happened in QG. Is it clear? From this, we can say that recombination has happened in the Q gene. Now, if you have three genes, suppose PQR, let's say PQR, then this question will be of a 3 point test cross. And if they ask you, suppose, and from here you got to know that recombination has happened in RG, by just like this comparison if you do, you got to know that recombination has happened in the R gene. So what does this imply? This imply that R is present in the middle. The order is PRQ. So for 3 point test cross, always remember that the gene that shows recombination is the gene that is present at the center. Okay, so suppose if you have to, if there is a question where you have to uh, determine the exact sequence of the gene, in that case you have to see that which gene is actually crossing over between your recombinant classes and parental classes. So let's say if your R gene is is showing crossing over between your recombinant and parental class, that means R is present in the middle. So gene which shows crossover is present in the middle. Okay, this is only for 3 point test cross, but it is an important point. Gene that shows crossing over is present in the middle. This is for 3 point test cross. Okay, now like in the question, now we can solve this question easily. We have to calculate the recombination frequency based on two-factor analysis. This is a simple question. It can be just solved very quickly by applying the formula. Why I am spending so much time and deliberately? Because I want you to understand the concept, because the question might be a difficult version of the same concept. That's why you need to know the intricacies of how to solve this question. Okay. So what is the formula for calculating your recombination frequency? The formula for calculating the recombination frequency is recombination classes, that means classes that shows recombination, upon total okay, just let's write it like this. Hmm. Number of individuals that shows recombination upon total number of individuals into 100. Okay, so this is the formula for calculating the recombination frequency. So then what would be the recombination frequency? So which classes are showing here? 35 and 25, these are the recombinant classes. So it would be 35 plus 25 upon 350 plus 400 plus 35 plus 25 into 100. And when you calculate it, it will come approximately 7.4%. Okay. So because they are asking in frequency, so in numeric it is 7.4%. Okay. I hope this question is clear. Yeah, 3-point test cross. See, I know 3-point test cross is for order of genes. I was just explaining here. This is not a 3-point test cross. Okay, don't confuse it. I was just explaining a concept here that suppose if a question where three genes are there, I mean more than three genes are there. Okay, and if you have three genes and if you have to determine the sequence or the order of the genes, then how can you determine it? You can determine it by knowing that which particular gene amongst the three is getting crossover. So whichever gene is getting crossover, that gene is present at the middle. Okay, I told this 3-4 times that this is not applicable to this. Do not get confused. This I am telling you for 3-point test cross. I was telling, so for understanding how to uh, determine the gene which is getting crossed over, I did all this assignment, this explanation, that these are the wild types, these are the recombinant types, how you have to compare and find out which gene is getting crossover. Because the concept of genes that gets crossover, I mean the this is similar here. I mean how you have to find out which genes are getting crossover, like this only. Just the difference would be that there are two genes. In 3-point test cross there would be three genes. So amongst the three genes you have to see which gene is getting double crossover. There comes the concept of double crossover and single crossover, that which two recombination, I mean which two classes are showing double crossover. So just the comparison mechanism is similar. This order determination, see this is very but obvious. Just apply a little bit of general knowledge, that if there are only two genes, is there any point of determining the order or determining the sequence? No. If there are only two genes, P and Q, what order will I determine of this? That it's just two. If there are three genes P, Q, R, then only the question will come now whether the genes sequence is P, R, Q or Q, P, R or R, Q, P, whatever it is. So the question of determining the order will only come when there are at least three genes. With two genes, there is no order. They are just two. So this explanation is specifically for 3-point test cross. But to determine which gene is getting crossover, I explained with the help of this 2-point test cross example. Okay, do not get confused in this. Okay. Now, next question, again, a very interesting question. Hmm. Consider the following pathway and identify the correct statements. Okay, this is the pathway given to you. APAF is epistatic to back and backs. APAF1 is epistatic to cytochrome C. BCL2 APAF double mutant cells will not undergo apoptosis. Cytochrome C APF1 double mutants will not undergo apoptosis. A gain of function mutation in Cas9 may display the same phenotype as BCL2 deletion. A loss of function mutation in BCL2 will display the same phenotype as Cas9 deletion. So, I will give you 3 minutes to solve this question. Think logically, carefully. If you have gone through uh, uh, the lectures, the recorded videos, then you will be able to answer definitely. Think and try to answer it. 3 minutes for this question. One more minute. Okay, now I'm switching the screen. Hmm. So Ajay has answered only last option is incorrect. Hmm. So anybody else who would like to try? Jasreena, Kartika, Shubhamoy, can you understand this question? Try to solve this question. See what they are asking. They are giving a pathway, okay? So it's a pathway you can see. BHK, BH3, BH3 giving rise to, BH3 increases expression or give rise to BAK backs, then cytochrome C, then APF1, then Cas9, and ultimately apoptosis. So it is the pathway of programmed cell death or apoptosis, what we say. So and in between BCL2 inhibits in this process, in this pathway, it shows that this is the general mechanism through which apoptosis occurs, and BCL2 it inhibits the step where back and backs forms or induces cytochrome C. So this step is inhibited. So this is the question, and according to this question, these are the some statements that are written. Okay. So now, can you think what can be the answer? Anything that you can think of? Okay. So can anybody explain me what is the meaning of epistasis? Write it in the message box. Anybody who knows what is the meaning of epistasis? Because two things are important here. First is yes, yes, Shubhamoy, you wanted to say something? Hmm. A produces mutation. Okay, fine, Shubhamoy. Okay. So if you do not have any query, then you can uh, turn off that raise hand because I'm getting confused every time I thinking that you're asking something because you have raised hands. Yeah, fine. Okay. So now, so Ajay has written A produces mutation, AB also produces mutation, but B cannot produce mutation. I hope that means A comes upstream. Okay. So I think Ajay has read the class because sir has also explained in a similar manner. I hope I have read it correctly because you have written many spelling errors, but I think this is what you meant. So what Ajay is saying that A produces a mutation and AB alone can produce a mutation, but B cannot produce the mutation. That means A comes upstream. Okay. What Ajay has told that I will explain. First, I will explain it in my way. Okay. So my idea here is that doesn't matter we are solving 10 questions or 15 questions or 20 questions. There is no point of solving 30, 40 questions. But if we are not able to understand the concept, just going through the questions and answering will not make any difference for your concept building. So it's better to solve fewer questions, but at least try to understand the sorry, but at least try to understand the concept behind it. Because when you are able to understand the concept, these are very important concepts, okay? And these are the concepts which are not given in your textbooks. They can only be explained to you. For example, 3 point and 2 point test cross. No matter n number of textbooks you read, n number of language you read, unless and until someone is explaining you on the board, it's very difficult. Okay. So Ajay, you want to say something? Okay, I think you wanted to say what is sir explained with car manufacturing line. Yes, yes, sir explained with car manufacturing line. That is, yes, that's correct. Okay, so now we'll start this question by
Madam,
first understanding. Uh, yes,
sorry.
Ajay, you want to say something? You can say.
With this question and that question number six, I think hypermorphic dominant gain of function. If there are multiple options, which option we should choose?
You can choose multiple options. So generally in MCQs there are one option only. So I have done
Hmm.
other NPTEL courses also in past. So they were just one options. But I think in Sir's course, Sir has given in framed questions in such a way that there are more than one options correct. So in your MCQs, if there is a possibility, I don't know how he's going to frame the questions, but if there are more than one correct option, and if your computer screen or whatever, I don't know whether it is a offline or online exam. If it is an offline exam, you encircle all the correct options. That is not a problem. You can encircle more than one options. If it is an online exam, then I don't know what how the software will allow. And this question, I think, in after some time, there would be a live session with your instructor also. Course, sorry, just 2 minutes. Yeah. So I think there would be a live session with your course instructor also, where sir will only come and have a live session with you all. So there you can ask this question that sir, like in like in live lectures, there were many questions which has more than one options, correct? So if such question comes in examination, should we encircle one or should we encircle all the correct options? And also you can ask one more question, just write it down for your own benefit, that what if you have answered three options as correctly, two of them are correct, one is incorrect. So whether you will get marks in that case or not, or whether how much marks would be deducted. Could you understand what I'm trying to say? Suppose if there are five options and three of any three of them are correct, and you have chosen a combination where two of your questions, I mean two of your options are correct and one is incorrect. Then in that situation, how what would be the marking scheme? You can ask him. He would only be able to tell you the best way how to answer such questions. Okay. Zero, I hope it is not zero, because two you have answered, so you can ask him, or you are writing your examination, I mean assignments from there. I think you might know now how is the markings scheme. Anyways, so let's come to this question. So we are starting with epistasis. Okay. So epistasis is a circumstance, is a condition where the expression of one gene is modified by the other gene. Okay. In very basic language, in very basic terms, if I explain what is epistasis, so this would be that expression of one gene is modified by another gene. Now, this modification, it can be a masking effect, that one gene is masking the expression of another gene, or inhibiting effect or suppressing effect, anything it can be. Okay. So this is what epistasis is, that one gene is uh, dependent, the expression of one gene is dependent on the expression of another gene, or the what would be the ultimate outcome, it depends upon the how the two genes are interacting with each other. Okay. So one gene can modify the expression of another gene. So the expression, the ultimate expression would be, would depend upon the presence or absence of the second gene. Okay. Now, if you uh, if you remember, we have studied about Mendelian genetics. So, what do you think, that this epistasis follows Mendelian laws of genetics, or it does not follow Mendelian laws of genetics? This epistasis, does it follows Mendelian law of genetics? No. Good. Jasrina has very correctly said, and Ajay also, it doesn't follow. Because Mendel said that each gene is getting inherited independently. There is no association between the genes. But in reality, it is not true. In fact, some genes, they do show interaction, and the expression of one gene is dependent upon the interaction that it receives from another gene. Okay, so therefore this is like whenever you study genetics, whenever you start genetics, first thing that you study is the Mendel's laws of inheritance. After studying Mendel's law of inheritance, then you start to studying the divergence, some applications or some phenomena which diverge from the Mendelian laws of inheritance. And the first thing that you study is epistasis. Okay, so now we know that this what is the meaning of epistasis. Now, very briefly, if you look into this question, just a minute. So this pathway represents an apoptotic path. Okay, so I'm not going to explain the whole pathway because it will take another 15 minutes for me. We have to solve other questions also. But in just in brief, what I can say that this BHK, it's a pro-apoptotic gene. Pro-apoptotic genes means that BHK, it promotes or it supports apoptosis. Then your BAK and BAX, these are also pro-apoptotic genes. These all are pro-apoptotic genes. And this BCL2 is an anti-apoptotic gene. Okay, this is anti anti-apoptotic gene. That means when BCL2 comes, it stops the apoptosis process. But you see in this figure that this figure describes the situation that is actually happening. That BH3 is getting converted to BAX, BAX is getting converted to cytochrome C. But what is happening that BCL2 here it comes and it stops the process. But even then, APF1 is able to convert Cas9, activate Cas9 and apoptosis is happening. That means even in the presence of BCL2 or BCL2, this APAF is able to show its function. Okay, what does it means? It means that APF has a very dominant role here. So APAF is able to mask this particular area, mask the expression of back cytochrome C or BCL2. So it is continuing the function of apoptosis. That means APAF is epistatic to back and backs. When I say epistatic to back and backs means APAF is able to mask the function of back and backs. Okay. Then APAF is also epistatic to cytochrome C because APAF1 is able to mask the expression or mask the function of cytochrome C. Because this particular portion, if this particular portion was really happening, then this apoptosis could not have happened because BCL2 is inhibiting the conversion of backs and cytochrome C. But because when APF is present, it masks the expression of back cytochrome C. So there is no effect of this particular step because APF is dominant over backs and cytochrome C. So this is correct. This is also correct. Now comes the next point. BCL2 and APF1 double mutants will not undergo apoptosis. That means if BCL2 is mutated and APF1 is also mutated, then it will not undergo apoptosis. So you can say because APAF1 is epistatic to back and backs. Okay, so if BCL2 is mutated, okay, it can go from here to here. This step can be passed. But because APF1 is also mutated and APF1 is dominant over back and backs, it masks the expression of back and backs. So if APF1 is mutated, then the further steps cannot happen. So this is also correct. That is BCL2 and APF1. Just give me 1 minute. I'm speaking continuously. Okay. Okay, fine. So, because APAF1 is the central factor which is responsible for apoptosis here. Central factor. Because APAF1 basically, okay, I'll explain it later. So, this is also correct. Then, cytochrome C and APAF1 double mutant will not undergo apoptosis. Obviously, see, if APAF1 is mutated, apoptosis cannot happen. If cytochrome C is mutated, then also, if only cytochrome C is mutated, then apoptosis can happen because APAF1 is epistatic to cytochrome C. But because APAF1 is also mutated, so apoptosis cannot happen. Correct? Okay. Then next option is again, hmm, a gain of function mutation in Cas9 may display the same phenotype as BCL2 deletion. Yes. Why? Because if BCL2 is deleted, then what would be the consequence? Apoptosis will occur. And if Cas9 is also getting hyperactivated, the phenotype would be apoptosis will occur. So both will show the same phenotype, correct? Then a loss of function mutation in BCL2 will display the same phenotype as Cas9 deletion. Will it show the same phenotype? No. Why? Because if BCL2 is lost, then it will support the apoptosis process because BCL2 is inhibiting it. But if Cas9 is deleted, then apoptosis cannot occur because Cas9 ultimately initiates a protein cascade and cleaves the target protein. It is a downstream of APF. So this is wrong. So all the statements are correct and the last one is wrong. Okay. So in these kinds of questions, is it uh, okay? Could you understand? So I have explained it, but you yourself have to go and read this question at least 2 to 3 times to understand each and every step. Okay, so Jasrina doesn't follow. So can you tell me, Jasrina, what step you couldn't follow?
Madam, she had answered you the Mendel's law don't follow. And myself also
Oh, acha,
answered
acha,
it would.
acha, acha. Okay, 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 okay. So thing is, huh? So this is your thing. Then there, huh. So Ajay, any term for unprogrammed cell death? Yes, there is a term, and that term is mostly non-programmed cell death represents a phenomena which is necrosis. Okay, so there are two terms: programmed cell death, which is apoptosis, and unprogrammed cell death is necrosis. Okay, so necrosis basically observed in some kind of traumas or any kind of stress conditions. So it happens and it causes the degradation. Okay, and yeah, I think this much is enough for your question. You can go in detail and study about necrosis, but unprogrammed cell death is necrosis. Now what I wanted to explain over here is, so from this diagram or from this flow chart, one thing is important, then APAF1 is very important. Okay, we can get to know that, yeah, we get to know that APAF1 is important. So basically, you remember that APAF1 it oligomerizes. just little bit knowledge, and it forms apoptozoa and forms apoptosome complex. And this apoptosome complex, what it does is, every time I lose my, it activates procaspase 9, and then this procaspase 9, it initiates a protease cascade. Okay, every time I lose my cursor, I don't know where it is now. Yeah, it activates a protease cascade, and it will, this protease cascade, it cleaves your target protein, which ultimately leads to cell death. So this is why this APAF1 is important in the apoptosis process. Okay. Yeah, so this is the answer. We have already. Now, another question of similar type. Consider the following pathway and identify the correct statements. So, this is a pathway which is given to you, and the statements are: synthetic phenotype would be observed in RAF MEK double mutant. Synthetic phenotype will be observed in a JAK2 STAT3 double mutant. Okay. Synthetic phenotype will be observed in a MEK STAT3 double mutant. Synthetic phenotype will be observed in a JAK2 ERK double mutant. Gain of function in EGFR will cause excessive proliferation regardless of IL-6 mediated induction. Gain of function mutation in EGFR will cause excessive proliferation only if IL-6 is deleted simultaneously. So this is your question. Try to answer it. I will give you 3 minutes. So it's right now it's 8:28. So 8:31 I'll switch the screen, okay? 3 minutes is more than enough for this question. Try to answer it, otherwise I'll explain it anyways. Think of all the options carefully and see what can be the correct options. Try to think, try to see which phenomena, just as a hint, which phenomena is working here. Okay, I'm switching the screen. It's 8:31 now. Yeah. Hmm. See whether you are able to answer this question or not. Okay, good. I think students have answered it. Not visible properly. Oh, I didn't see. Do you want me to show this question again? I can do it in a uh,
No, madam. Thank you.
okay. So C, D, and F. C, D, and F options. Just a minute. Actually, it's very small. Hmm. C, D, and F. Okay. Hmm. And Kartika has answered E. Okay. Okay. Synthetic phenotype would be observed in MEK STAT3 double mutant. Synthetic phenotype will be observed in JAK2 ERK double mutant, and gain of function in EGFR will cause excessive proliferation. Okay. Good, Ajay. So, this question essentially involves the concept of synthetic phenotype. Okay, so if you have gone through sir's lecture, then he has explained in detail about what is a synthetic phenotype. So in a very easy language, I'll tell you that when two mutations are combined and it leads to a phenotype that is different from the phenotype of either mutation alone, that is a synthetic phenotype. Okay, I'm again repeating the sentence. I'll write it also. When two mutations are combined and it leads to a phenotype that is different from the phenotype of the either mutations alone, that is known as a synthetic phenotype. Okay, what it says? It says when two mutations combined and it leads to a phenotype, uh, it leads to a phenotype that is different from the phenotype of either mutation alone. Okay, this is your synthetic phenotype. I hope, I'm sorry, my handwriting is not very good, and especially when I write through this tab, it becomes even worse. Anyways, so now we got to know that in this question, which concept we have to apply? We have to apply the concept of synthetic phenotype. And what is the definition of synthetic phenotype? That when two mutations are combined and it leads to a phenotype that is different from the phenotypes of either of the mutations alone, that is known as a synthetic phenotype. Okay, now the question comes, how to solve such questions? how to apply this concept of synthetic phenotype in these questions. So, to apply, there is one very important uh, uh, characteristic of synthetic phenotypes that you must always remember, and that is that synthetic phenotype is more likely when the two components of different pathways are involved. When components of different pathways are involved. I am telling you in context of this question
The
that
screen is not visible. Screen is not.
uh, screen is not visible. Oh, how come?
How
Just a
visible?
minute. Is it visible?
Yes, madam.
Okay, fine. So, hmm, a synthetic phenotype is more likely when two components of different pathways are involved. Okay, pathways are involved. This is important, and this line will help you to solve the question. Then two components of different pathways are involved, then a synthetic phenotype is more likely to occur. Now, why synthetic phenotype is more likely to occur when two components of a different pathways are involved? Let's see here. See, is the screen visible to everybody? Is the screen visible? I hope it is visible because I have switched to the presentation mode. I hope the screen is visible to everybody because I am not able to see in the message box. Otherwise, I have to, because for that I have to switch the screen. Now you see, if I talk, this is one pathway, this is another pathway. This is EGFR, which is basically upstream of the pathways. It is upstream of both the pathways, or at least to this pathway, it is upstream of it. Okay. Now, if I said that synthetic phenotype is more likely when components of different pathways are involved. Suppose if RAF is mutated and MEK is mutated, then what will happen? RAF is mutated, so MEK is not generated, so cell proliferation will not take place. It is but obvious. Something different can come only when, if suppose RAF is mutated and let's say JAK2 is mutated or IL-7 is mutated, then some difference, I mean some interesting or contrasting characteristics can be occurred now. Because if the components of the same pathway are only mutated, then what is new in this? There is nothing new in this. It is but obvious. If RAF, which is the upstream molecule, if this is only mutated, then it will not activate MEK, it will not activate ERK or ERK, then cell proliferation will not happen. Nothing new, nothing interesting here. But something new interesting will come only when, let's say RAF is mutated and IL-6 is mutated. So whether these two, when combined together, are they producing any different phenotype? That is the thing. Suppose if there is a gene A which is responsible for cell cycle arrest, and there is a gene B which is responsible for checkpoint clearance, individual function of A and B, and these two are involved in different pathways. When these two genes are combined, they are producing some another third phenotype, anything, then that is a synthetic phenotype. So now, after all this explanation, if you go through the question, the first option says synthetic phenotype will be observed if RAF and MEK in a RAF and MEK double mutant. Just now we have explained So will there be any synthetic phenotype? RAF and MEK, they belong to the same pathway. So no, there will not be any synthetic phenotype. Then synthetic phenotype will be observed if JAK2 and STAT3 in a JAK2 and STAT3 double mutant. Again, will there be any synthetic phenotype in this case? No. Why? Because both JAK2 and STAT3, they belong to the same pathway. This is also incorrect. Now come to the third option. Synthetic phenotype will be observed in a MEK STAT3 double mutant. Now that is something we can see because MEK is belongs to a different pathway and STAT3 belongs to a different pathway. So if double mutant means what? Double mutant means both MEK and STAT3 are mutated. Suppose if there was written in a JAK2 STAT3, sorry, MEK STAT3 single mutant. If single mutant is written, then it will not show a synthetic phenotype. Always remember. It will show only when it is a double mutant. That means at least two mutations have to be there in two genes. If there is just one mutation and other gene is functioning normally, then it is not a synthetic phenotype. Synthetic phenotype when you are taking two genes of two different pathways and both the genes are mutated. Okay, so this is correct. Then next is synthetic phenotype would be observed in a JAK2 ERK double mutant. JAK2 and ERK. Double mutant also, different pathways also. Correct. So these four options were easy. Now comes the the last two options. Okay. This says gain of function mutation in EGFR will cause excessive proliferation regardless of IL-6 mediated induction. Will it be correct or not? Yes. Why? Because EGFR is upstream of IL-6 or and RAF. So EGF, whatever is upstream, is not dependent on the downstream molecules now. The expression of EGFR is not dependent on IL-6. So whether IL-6 mediated induction is happening or not, the EGFR function will continue. It is independent of IL-6 function. Okay, because it is upstream of it. So this is correct. Gain of function mutation in EGFR will cause excessive proliferation only if IL-6 is deleted simultaneously. No, because just now I said that EGFR is present upstream. So it is not dependent on IL-6. There is no dependence of EGFR on IL-6, neither a positive dependence nor a negative dependence because it is upstream of it. For example, if A gets converted to B and B gets converted to C. So B is dependent on A because B will get activated only when A is there. C is dependent on B also, C is dependent on A also. But is A dependent on anything? No, A is not dependent on B and C. A is A is inducing the expression of B and C. A is not dependent on B and C. So similar concept applies here also. That EGFR is the uppermost molecule. It's the first molecule, so it is not dependent on IL-6 function. Whether it is a positive dependence or negative dependence, that is not the question. Okay, so this option would be incorrect. So these three options, if this is A, this is B, C, D, and E options are correct. Okay. Okay. Now we'll come to the last question. Which of the following statements regarding vulval development in C. elegans are correct? Laser ablation of anchor cells has the same effect as Lin3 deletion. Lin3 deletion results in a hypodermal fate of all the VPCs. Overexpression of Lin12 in central VPC will have no adverse effect on vulvar development. Laser ablation of tertiary VPC inhibit vulva formation via Lin12. Lin12 acts as a negative regulator of central VPC fate. Loss of Lin12 induces all cells to form hypodermis. This is a question. This is not, I mean, there is nothing much to explain, but I'll try to explain just the basics. So, for this question, you need to have a basic knowledge of vulval development. So, if you know how vulval development takes place, then it would be easier for you to solve this question. Okay. So, even if you cannot answer it, you should go and read about vulval development. So, that will help you to solve this question. But right now, I'm giving you 3 minutes to solve this question. It is 8:47 right now. At 8:50, I will switch the screen, then I'll see what you have answered, and then we'll try to discuss this question in the last 10 minutes. Okay. So now try it. Try to, I mean, just go through the options carefully and then try to answer it. Okay. So you have 1 more minute. Try to answer the question. Otherwise, anyways, I'm going to explain you. Try to, if you know the basics of, I mean, vulva development, then you will be able to answer this question. Is easy. Okay. Now I'm switching the screen. It's 8:50 now. Fine. Hmm. So, Jasrina has answered A and B. Any other? Ajay, could you answer this question? Acha, you have answered. Okay. Okay. Fine. So let us see. So, starting with some background information. So, as I already told you, that in C. elegans, so the development of vulva, it involves a series of interactions, okay? And these interaction occurs between your anchor cells and vulval precursor cells. And basically, lin 3 and lin 12, they are important. Why they are important? Because lin 3 is a ligand that acts on the vulval precursor cells and it induces the vulva development. So lin 3 is important for vulval development, and lin 12, it is important for lateral inhibition. Okay, so lin 3 is responsible for vulval development. So the central region receives the highest amount of lin 3 and it becomes the primary cells. And then the adjacent numbers in the adjacent ones, they receive little bit less and they become the secondary ones. And then the later ones who does that does not receive the lin 3 are hypodermis. So now, in accordance with this, let us try to understand each and every uh, statement. Okay, so the first statement says that laser ablation of anchor cell has the same effect as lin 3 deletion. So basically, like we said, that C development of C elegans, it involves as a interaction between anchor cells and VPC, vulval precursor cells. So anchor cells are the cells that produces lin 3. Okay, anchor cells produces lin 3. So if laser ablation means through laser you are trying to you are destroying the anchor cells. So if your anchor cells are destroyed, then what is happening? The source of lin 3 is also destroyed, because anchor cells are only producing lin 3. So laser ablation means that through laser you are trying to destroy the anchor cells. So if your anchor cells are destroyed, your lin 3 production is also halted, because anchor cells are only producing your lin 3. That means laser ablation of anchor cell has the same effect as lin 3 deletion, because laser ablation of anchor cell actually leads to lin 3 deletion. So this is a correct statement. Okay. Now we come to the next statement, which says lin 3 deletion results in hypodermal fate of all vulval precursor cells. It is true, because lin 3 only determines it would be primary, secondary and tertiary. Okay. Lin 3 is the induction signal. Okay. So when there is no lin 3, none of the VPC will receive the induction signal, and they will adopt all the precursor cell, vulval precursor cells, they will adopt the non-vulval fate, which is the hypodermal fate. So if there is no lin 3, there is no induction. There is no induction means all the primary precursor cells will become the non-vulval cells. So this is also a true statement. Second is, overexpression of lin 12 in central VPC will have no adverse effect on vulva development. Is it true or false? It is a false statement. Why? Because lin 12 is responsible for lateral inhibition. Okay, what does it means? That lin 12 ensures that only one VPC adopts the primary fate, while others adopt the secondary or tertiary fate. What does it mean? That suppose these are the precursor cells, and if lin 3 is there, now these three, suppose these two cells are, rece- are receiving approximately equal amount of lin 3. So can both the cells become primary cells? No. Only one cell can become primary cell. So to stop the other cell from becoming primary cell, Lin12 plays the role. So it causes lateral inhibition through which it ensures that only one cell becomes the primary vulval cell. Okay. So when there is overexpression of Lin12, it will disrupt the normal lateral inhibition process. So there would be some abnormal vulva formation, it can be there. That means, uh, so it will not be a normal condition. There would be some or the other kind of abnormal vulva formation condition. So this is a wrong statement. Then laser ablation of tertiary VPCs inhibit vulva formation via Lin12. This is also a false statement. Why? What it says is laser ablation of tertiary VPCs inhibit vulva formation via Lin12. So, tertiary vulval precursor cells, they normally adopt the hypodermal fate. Hypodermal fate means the non-vulval fate. Okay. So, they do not play any direct role in vulva formation. So if even if you are uh, disrupting your tertiary VPCs, there would not be any effect on vulva formation. Remember this. Why? Because your tertiary VPCs, they do not play any direct role in vulva formation. So ablating them, they would not have any, I mean, it would not have that effect on vulva formation via Lin2. So this is false. Then Lin12 acts as a negative regulator of central VPC fate. Yes, it acts as a negative regulator. Why? I already explained that it causes lateral inhibition. That means it helps that only one of the vulva precursor cell, it adopts the primary vulval fate, and other precursor cells will adopt the secondary or tertiary vulval fate. So this is correct. Loss of Lin12 induces all cells to become hypodermis? No. What will happen? If there is no Lin12, then there would be no lateral inhibition. Okay. So actually it loss of Lin12 will actually have the opposite effect. So there would not be any lateral inhibition. So there can be a possibility that more than primary vulval precursor cells, more than one primary vulval precursor cells can be there. So it will be an abnormal condition, not a hypodermis condition. Okay. So this is also a wrong statement. So from here, what can we see? First, A is correct, B is correct, E is correct. Yeah. Huh, this is also correct. Okay. So with this, overexpression of Lin12, what I said, overexpression of Lin12 in central over, this is also correct. Sorry, it is correct. C is also correct. Why C is correct? C statement says that overexpression of Lin12 in central VPC will have no adverse effect on vulva development. Why? Because Lin12 in the central VPC, because in the central VPC, if Lin12 is expressed, then what will happen? It the function of Lin12 is to uh, cause the lateral inhibition. So if it has caused the lateral inhibition, then apart from central vulva, if your primary VPC is determined, then after that, whatever it is happening, it will not have that much effect on vulva development. Important is that at least one of the primary vulval precursor cell should be determined to form the vulva. That is most important thing over here. So yes, C is also correct. So we have A, B, C, and E as the correct options. Okay. So just a minute. So with this, we complete the all the questions of today's week lecture. And within next, so next week, I think next week class would be at the same time, Tuesday, sorry, Monday, 6 to 8. This time because I was not very well. That's why today also I'm not very well, but I had to take class anyways. So now we'll meet next week, same time, like Mondays, 6 to 8 p.m. Okay? Chalo. Thank you, everybody. Bye. Have a great week.